Bismillahirrahmanirrahim. Now this is another video on the paper 4, which is your first exam this coming year, and you will be doing this exam very soon. So I think I will post a few more videos on this so that you all can improve your skills in this. As you all know, this paper is for 2 hours, and this is a very difficult paper. And we have to also understand you have to answer all the questions. Previously there used to be a choice, and it is for 100 marks, and there is no section B, which means there are no essay questions. So there are no essay questions since 2022 when the new syllabus came in. So no essay questions, no section B anymore. Now the first thing which I want to educate you with is the grade thresholds. Now in this paper, and if you look at the grade threshold, now you can see here in paper 4.1, what we have to understand is how it's going to happen 4.1, 4.2 and 4.3. Of course we have 4.1, 4.2 in Pakistan. Now 58 out of 100 was an A grade and 49 was a B grade and 40 was a C grade and 31 out of 100 was a D grade. So 21 was an E grade, which is just a pass. Now I always say that getting even 60 out of 100 is a pass. So you look at all the 4.1s and 4.2s and 4.3s, out of 100 it's 60, 58 and 68. So anything between 60 and 70 out of 100 should get you an A grade. So please do not spend time on the questions that you do not know. You might do a paper, only a 90 marks paper, and get 60 out of that and leave out 10 marks of questions. But then what have you done? You have done it sensibly. You're sure to get the 60 out of 90 or 60 out of 100 marks. So you've not done 10 marks of questions. You have just left them because you didn't know them. But the ones you knew, you did it so well that you have been managed to score 60 out of 100. But you only did a paper worth 90 marks. So you didn't do a question, or you didn't do part of a question, or you didn't do a part of 2 questions. Now let's start with the question number 1. There's no choice. You have to do all the questions. The golden mantella, whatever, is a small terrestrial frog found in Madagascar. Figure 1.1 shows a golden mantella. So you can see this very nice looking frog. Name the domain and kingdom to which the golden mantella belongs. Do you know the domain is eukarya? What are the domains? There are 3 domains. There are 3 domains. What? Archaea, bacteria, and eukarya. Archaea, bacteria, and eukarya. Always make a mnemonic to remember things. So, and the kingdom is Animalia. Because it's an animal, frog is not a plant, it's an animal. We all seen frogs, so it's not something that we haven't seen. So, this got you your 2 marks. The skin of the golden mantella is brightly colored and contains a toxin. Suggest a benefit to the frog of being brightly colored. The skin of the golden mantella is brightly colored and contains a toxin. Suggest a benefit. So they're giving you something for you to think about. Why brightly colored? And then, so it's a warning to predators, or you can say to deter predators. So that was to keep away the predators which are going to pounce on it, because it contains a toxin which is going to kill the predator. Now, whenever there is this thing written at the mark scheme, it means any valid point. Like, for instance, somebody might have written brightly colored, so is very well camouflaged in the background, or maybe in the flowers and the plants which are very brightly colored. So it's very well camouflaged, so is not easily predated. So AVP means any valid point, and this of course can be, we can always discuss this when, as if I'm the examiner, then we talk with the head examiner and then we say, okay, well, one student wrote this, what do you think? And if that is a relevant point, then we will give that person a mark. Then coming to the B part, the toxin in the skin of the golden mantella affects the action of the sarcomeres in the muscle fibers of mammalian striated muscle. The toxin inhibits a protein calcium ATPase found in the membrane of the sarcoplasmic reticulum. Calcium ATPase pumps calcium ions from the cytoplasm into the sarcoplasmic reticulum, when the fiber is no longer stimulated. Suggest the consequences to the sarcomere of the action of the golden mantella toxin, and that's 3 marks. Now, naturally, the calcium stays in the cytoplasm, so the calcium binds. You see, calcium binds to troponin, tropomyosin shifted, and the myosin actin cross bridges remain, so the sarcomere muscles remains contracted or shortened, or you can say remains paralyzed. So you had to talk, if you knew the physiology of the muscle, of the striated muscle, only then you could have answered this question. This was a very, very direct question of if your understanding of the striated muscle was clear, only then you could have done this question. Now, if you look at the normal functioning of the muscle, striated muscle, you see, now I'm going to revise this with you. Acetylcholine is released from the axon terminal of a motor neuron and binds to receptors in the motor end plate. This binding elicits an end plate potential which triggers an action potential in the muscle cell. So what you have to understand is, this is the first point. Second, action potential propagates along the sarcolemma and down the T tubule. Number two. Number three, the action potential triggers the release from the sarcoplasmic reticulum, which is the endoplasmic reticulum, is called sarcoplasmic reticulum. Number four, calcium binds to troponin. So the action potential triggers the calcium release. The calcium binds to troponin, exposing the myosin binding sites. Number five, cross-bridge cycle begins. Number six, this is the important thing. Calcium is actually actively transported back into the lumen of the SR following the action potential. Number seven, tropomyosin blocks myosin binding site, muscle fiber relaxes. Now in the question they've said is, what if this calcium would not go back in? So step 6 is not going to take place. So if step 6 is not going to take place, then calcium will stay in the cytoplasm, which is also called the sarcoplasm, and the calcium will remain binding to the troponin, here this point here, and the tropomyosin does not cover the myosin binding site, so the myosin actin bridge exists, or they remain, and the muscle becomes paralyzed. So this is what you have to understand. If you have understood this, then you could have done that question as well. Then coming to the C part of the question, describe the role of calcium ions in a cholinergic synapse. Very basic question, very direct question, no ambiguity about it. Calcium enters into the synaptic knob. So this is the synaptic knob, and there is a calcium channel, and calcium is always outside more, so it will enter and then it will be actively transported out again. So calcium diffuses into the synaptic knob through voltage gated calcium channels. Through voltage gated calcium channels, then the vesicles containing acetylcholine, there are vesicles here which contain acetylcholine. So the vesicles containing acetylcholine will move and fuse with the presynaptic membrane, and then they photophy, as a joke, exocytosis. Exocytosis means all these acetylcholine is going to be passed out. So exocytosis. Now, I've explained this to you. This is a very direct question. This comes many times in the papers. If you look at many old papers, this come many, many times. Now, as you can see, I've written the answers to this. Calcium diffuses into synaptic knob, 1 mark. Through calcium ion channel, 2nd mark. I mean, all of you knew that, but you won't write it, I know it. Vesicles containing acetylcholine. Now, the parts which I've underlined in red means those are essential. You had to give this word. You couldn't have come up with anything else. You couldn't have said vesicles containing neurotransmitters or vesicle containing chemicals. The mark, in order to get this mark, you have to write vesicles containing acetylcholine. Move and fuse with the presynaptic membrane. Again, presynaptic membrane is underlined. And then exocytosis is underlined. So exocytosis is a word which we have to use. You can't say acetylcholine is just thrown out, or the vesicles burst and the acetylcholine just diffuses out or moves out. That's all rubbish. That is not going to get you any marks. You have to be very clear in writing the correct biological English. Now, coming to question number 2, in eukaryotes, the cells of plants and photosynthetic prototists contain chloroplasts. Describe the relationship between the function of a chloroplast and its structure. So you've got to give me points about the structure and its function. So of course you can come up with a whole lot of things, but be sure that you get your 4 out of 4, because this is one question which you can get your full marks. So there's the thylakoids. What happens there? Light dependent reaction takes place there. Thylakoid membrane, they contain the photosystems. The thylakoid membranes absorb light. The stroma is for the Calvin cycle. The stroma is for the enzymes like Rubisco and RUBP and reduced NADP. Then the stroma contains water so that the light reactions or as a medium of reaction for photolysis. Then the DNA makes the chloroplast proteins which are needed for photosynthesis. Then the starch grains are the store or the products of photosynthesis, which is the chemical energy. And so there were about 8 or 9 mark scheme points, and you could have given me any 4 of those. So be careful how you write this, and you can even do a diagram and write it. Even though there are lines, you can do a diagram on top of that. So you can easily do this part of the question. Now, the next part again was a very direct question. Outline the main features of cyclic photophosphorylation. So in cyclic photophosphorylation, what happens is light excites electrons in PS1. Electrons are passed along ETC, hydrogen protons pumped into the thylakoid, protons diffuse through the ATP synthase, and this results in the formation of ATP. Now you could easily have made 2 columns for structure and function, but it's up to you. You can always do a column, you can do without a column and just think like that. So the thylakoid is a structure, it is needed for the light dependent reaction. The thylakoid membrane have photosystems or ETC or ATP synthase. They're actually embedded in the thylakoid membrane. Then the grana is a large surface area so that it can absorb more light. Then the stroma is for the Calvin cycle. The stroma contains the enzymes RUBP and the enzymes required for the Calvin cycle. Then the DNA and the ribosomes are essential for making the proteins for photosynthesis. For all the reactions which are taking place in the chloroplast, it doesn't have to import those enzymes from the cytoplasm of the cell. So it can make its own proteins because it contains DNA and the ribosome. So here transcription and translation will take place. But there can be no post-translational modification because there is no Golgi apparatus inside the chloroplast. Then coming to the again direct question, light excites electrons in PS1, electrons pass along ETC, hydrogen ions pumped into the thylakoid space, and hydrogen ions diffuse through the ATP synthase, and this results in the formation of ATP. So light main features of the cyclic photophosphorylation. So there is a non-cyclic and there is a cyclic photophosphorylation. But in the cyclic photophosphorylation, only PS1 is involved. And the PS1, the electrons are excited, they come back through the ETC, and that results in the formation of ATP. I'm just going to give you a diagram to understand this. You can see this is a very good diagram showing you the light dependent stage in which we have the photosystem 1 and we have the photosystem 2 here, which is in the beginning, and then you can see how the red arrow is showing you how hydrogen ions, you see the hydrogen ions were where? They were outside in the stroma. This is the stroma, and they have been pumped in. Now you see this arrow, which I'm just doubling it up, and they've been pumped into what? Into the thylakoid space. And once they're inside the thylakoid space, now there are lots and lots of hydrogen ions here inside. Now there's only one way out for them. There's no other way out because please remember it's a phospholipid bilayer. Ions cannot cross it. So the only way that they can come out, they can go is through the ATP synthase. And when they pass through the ATP synthase, that's when ATP is made. And this ATP is going to go and be used in the Calvin cycle, which is going to take place in the stroma of the chloroplast. So these hydrogen ions, the story that I just did, that the hydrogen ions are pumped into the thylakoid space. Please understand, this is the stroma. This is the stroma here. And from the stroma, the hydrogen ions are, this is actually in the new syllabus. And this is what you must know very clearly, that we're going to be asking you questions on that, maybe in this year's paper as well. Now coming to the C part of the question. Now this is a question which you need to do with great care and concern. Why I'm telling you that is because it's very easy to get this marks in this paper, but if you do it very carefully. Red algae are aquatic protists that are multicellular. The cells of red algae have chloroplasts. Containing photosynthetic pigments, many red algae live in deep water. Read the question at least twice. Two of the accessory pigments of red algae are phycoerythrin, appears red, often present in large concentrations. So phycoerythrin which appears red, and phycocyanin, which appears blue. The first few meters of water nearest the surface absorb the red wavelength of light. So there is live in deep water. So I mean there is this whatever pond or water. So the first few meters of the water near the surface absorbs red wavelength. So this is red wavelength. If the water also contains particles of organic material, it absorbs blue wavelengths. If there is particles of organic matter like dead tree, dead leaves, dead animals in it, that will absorb blue wavelengths as well. Explain this to yourself. Figure 2.1 shows absorption spectra of some pigments in the red algae chloroplasts. So we have chlorophyll A, phycoerythrin, and phycocyanin. Now, what you need to do is you need to look at it very carefully. And now look at chlorophyll A. Now look at the pigment for chlorophyll A. I'm just going to color it and so that everybody is very clear about what am I going to talking about. Now I know you don't have colors there in the exam, but you can always do something to understand it. So it has two peaks. This is what? Chlorophyll A. Then we have phycoerythrin. Now phycoerythrin is this dotted line, so I'm going to use a green color for this. And you can see it just waxes and wanes and that finishes off here. So it is between 400 and 600. While the red was from 300 to 700. And so we have phycoerythrin, right? And then we have, let me use an orange color for that. And then we have phycocyanin. And where's the phycocyanin? It starts here, it goes up here, it goes up here, and then so it is between what? This area and this area, between 450 and 650. No, not 650. It's more than 650. 660. So this is the one that you have to understand. Now, if you've understood this, now what do we have here? We have percentage light absorption, and here we have the different wavelengths of light, and then you have the different colors, violet, blue, green, yellow, red, and all that. Now let's go ahead and read the next part of the question. Describe the differences. Now this is describe is one in which you can get full marks. Describe the differences in the absorption spectra of the three photosynthetic pigments shown in figure 2.1 and explain how these differences help red algae to survive in deep water. So there is a describe and there is an explain. And a describe means no biology. So in the describe, you will only talk about which wavelengths were absorbed for chlorophyll A. So chlorophyll A, you can see, is the red part. Chlorophyll A is the red one. That is absorbed mainly between, the peak is at 430, but it's between 300 to 450, and then the peak is between 650 to 700. So those are the wavelengths that are chlorophyll A. Then which are the wavelengths for phycoerythrin? Phycoerythrin is the one which we is the one that we have talked about, is the green one. Phycoerythrin is between 410 and 610. So phycoerythrin, we have to identify the wavelength. So this is not something that is no biology. It's something which you just have to do from the graph. And then you have to identify for phycocyanin. Phycocyanin is between 450 and 650. So these are the wavelengths during which this is the pigment which is going to be absorbing light. Then chlorophyll has peaks mainly in blue and red. Chlorophyll A has peaks mainly in blue and red. You can see the blue red here, the blue here. So peaks in blue and red. Then chlorophyll, the phycoerythrin absorbs in blue, green, and yellow. So it's in blue, green, and yellow, all this area, because this is where the phycoerythrin is working. And then the phycocyanin absorbs in mostly yellow and red. So it's got the peak here, which is this part, and you can see it's the yellow and the red. So basically, describe is nothing something which you need to have known before in your. It's not something the teacher must have taught you. It's just a skill of answering question. Now, these were the describe, and there are 6 describes, and the total of 7 marks. So you could have got your 6 marks for writing all that. And explanations, of course, are go back to it. Explanations are, of course, the bio part. So red algae, deep water gets green and yellow light. Then chlorophyll A absorbs little green and yellow light. Then the phycoerythrin and the phycocyanin are the accessory pigments. They absorb wavelengths not absorbed by chlorophyll A. Then combined pigments absorb greater range of all wavelengths. Then increases this absorption of light will result in higher rate of the light dependent stage of photosynthesis. You see, when light is absorbed, that is going to result in a higher rate of the light dependent stage of photosynthesis. So so there's going to be more organic compounds and more growth. So more ATP, more NADPH, more Calvin cycle. More organic compounds means more growth, so more glucose, more protein. Now, as I've copied down the mark scheme here, you can understand why I've done this. You see, D means these are the describes, and there are 6 describes, and it says any 7 from. So there wasn't this that 3 marks for describe and 3 marks for explanation. So any 7 from these. So these are the describes, and then these are the explanations. Now, as an A-level student, you must know what is a describe and what is an explanation. Explanation means biology. So you had to really look at all these points. These were explanations. Red algae, deep water, so gets green and yellow light. Chlorophyll A absorbs little green and yellow light. Then phycos are accessory pigments. This is again biology, absorbs wavelengths not absorbed by chlorophyll A. Then combined pigments absorb greater range of all the wavelengths. More light absorbed, rate of light dependent stage increases or photosynthesis increases. So more organic compounds for growth. So this is all what is, these are the explanations. Explanations is biology, but description or describe is not biology. Now coming to question number 3, respiration is a process that results in the synthesis of ATP. The ATP can be used within the cell for energy requiring reactions and processes. There are 4 stages in aerobic respiration: glycolysis, link reaction, Krebs cycle, and oxidative phosphorylation. The ATP synthesized in respiration can be used to make larger and more complex biological molecules from smaller molecules. Name the type of reaction that occurs when large, more complex biological molecules are made from smaller molecules. Well, that's easy, condensation. If you just said condensation, well, you got a mark. But if you said anabolic, even then that was correct. And you could also have said polymerization. So anabolic was right, condensation, polymerization. Then the first part of glycolysis uses ATP. Explain why ATP is needed in the first part of glycolysis. These are very direct questions, and everybody must know that it stops glucose leaving the cell, or you can say to phosphorylate or add phosphate to glucose, activates the glucose, and to make fructose 1,6 bisphosphate. So add phosphate to glucose. This was essential. You had to give this point to me. And then any one from the other stops glucose leaving the cell, activates glucose, or to make fructose 1,6 bisphosphate. Then coming to the C part of the question, state the precise locations of substrate-linked phosphorylation reactions in aerobic respiration. You know that occurs in glycolysis and the Krebs cycle. So glycolysis takes place in the cytoplasm. So if you said cytoplasm, you got a mark. And the mitochondrial matrix where the Krebs cycle takes place. Now, this is something all factual. You have to know this. You have to remember this. There is no way out. There are many things that you have to memorize. There's a lot of rata in biology. So, cytoplasm and mitochondrial matrix. Then coming to the deep part, explain what happens to pyruvate in the link reaction in aerobic respiration. It is decarboxylated, or you can say loses the carbon dioxide, and it's also dehydrogenated, and it forms acetyl. The 2-carbon acetyl group joins the coenzyme A to make acetyl CoA. Again, come many times in many papers. So decarboxylated, dehydrogenated, and 2-carbon acetyl joins coenzyme A to make acetyl coenzyme A or acetyl CoA. Then coming to the E part of the question, it's a total 9 marks question. Chemiosmosis is a process that occurs in mitochondria during aerobic respiration and in chloroplast during photosynthesis. Describe the differences between the process of chemiosmosis in mitochondria and the process of chemiosmosis in chloroplast. So in the mitochondria, it is oxidative phosphorylation. In the chloroplast, it is photophosphorylation. Now, similarly, you can do a little column here and you can write mitochondria and you can write chloroplast here. So you can do this. There's no rule that you can't draw a line here and you can't do a table here. You can do this like this. The in the mitochondria takes place on the inner mitochondrial membrane. In the chloroplast, it occurs on the thylakoid membrane. Then in the mitochondria, reduced NAD and reduced FAD give the electrons. In the chloroplast is photolysis. In the PS1, which gives the electron and the proton. Then in the mitochondria, there's a lot of hydrogen ions in the intermembrane space, while in the chloroplast, there are a lot of hydrogen ions in the thylakoid space. And in the mitochondria, oxygen is the final electron acceptor, and in the chloroplast, the NADP is the final electron acceptor. In mitochondria, it makes water, and in the chloroplast, it makes reduced NADP. Now, the important thing to understand in chemiosmosis is this is the diagram which everybody must remember very well in the coming exam. You see, there's a comparison between mitochondria and chloroplast. Now, in the mitochondria, in the intermembrane space, you see the hydrogen ions are more in the intermembrane space, while in the chloroplast, they are more in the thylakoid space. And this is the membrane which is there. And then, of course, what is going to happen is we have to understand is that the hydrogen ions will move from the thylakoid space into the stroma. So, thylakoid space into the stroma. While here, they will move from the intermembrane space to the matrix. So, there's a very lot of difference between the chloroplast and the mitochondria. In the mitochondria, they move from the intermembrane space to the matrix, while in the chloroplast, they move from the thylakoid space into the stroma through the ATP synthase molecule. And you can see this is the ATP synthase molecule and this is the formation of ATP. So everybody has to be very clear in this comparison. And you can see they have shown you from where they've taken it from the chloroplast and from where they have shown you this diagram from the mitochondria. So everybody must ponder about this. And I'm going to leave these diagrams at the end so that all of you can go through these and just have them, or maybe, just have them printed out or copied or look at them again and again. Now, the mark scheme of this is very clear. Chemiosmosis in the mitochondria is called oxidative phosphorylation. Here it's photophosphorylation. Then the mitochondria, the ATP synthase is on the inner mitochondrial membrane. The ATP synthase is on the thylakoid membrane. Then here reduced NAD and reduced FAD are taking the hydrogen for the ETC. Here there's going to be photolysis of water, and that is going to result in the hydrogen ions and the electrons, the water splitting into hydrogen ions and electrons. Then the hydrogen ions are on the intermembrane space. This is the diagram which I just explained to you. So they are in the intermembrane space. Here they are in the thylakoid space or in the lumen of the thylakoid. And then oxygen is the final electron acceptor. Here, no. NADP is the final electron acceptor and results in NADPH being formed, right? And here it results in the formation of water. Here it results in the formation of reduced NADP. So it's a totally different thing. That the comparison is a very nice comparison, and I hope this comes again in this year's exam or some sort of way so that you all can do it well. So please understand this part of the syllabus. Just giving you these diagrams once again, and you can just copy these down or print them out. This is another one very good, which is showing you how the hydrogen ions are moving in from the stroma into the thylakoid space. And then this is the diagram which I want you to know about the sarcomere and how the whole process is going to take place. I end this video here, and the rest of the questions will continue in the second video, which will follow this. Thank you once again.